Today our topic is experimental method. This is one of the method in epidemiology. So experimental method is third major approach, or you can say second major approach. Analytical method is the second one. So if it we see descriptive and after descriptive and analytical, then experimental is the third major approach, right? So it is distinguished by the active manipulation of exposure or intervention by the investigator with the goal of assessing its causal effect on health outcomes. Means we are not means we are able to see the manipulation of exposure and the casual effects whatever they have on the health outcomes by the experimental methods. So unlike observational studies, experimental studies introduce change under controlled conditions and observe the resulting impact. So this method is very essential for evaluating efficacy of the public health interventions. For example, like how the vaccines are working, how the health education programs are working, or environmental modifications are working on the health outcomes of the community. So in epidemiological experiments, the researcher manipulates the independent variable. He controls the study conditions, observes the effect on the dependent variable. So this method allows precise measurement, minimization of bias, and strong internal validity, making it ideal for the testing new interventions and confirming causal relationships. So experimental epidemiology as per WHO is defined as a study of relationship between exposure and outcome through controlled interventions where investigator assigns the exposure and observes the effect. And other related definitions, you can say an experiment is observation under controlled conditions, or experimental research is description and analysis of what will occur under carefully controlled conditions. So what are the elements of experimental epidemiology? The elements are like manipulation of exposure. We are or the investigator is actively introducing or modifying the exposure or intervention that is being studied. It can be a vaccine, it can be a medication, it can be a behavioral program, or any environmental change. Then we have a control group. Control group means a comparison group where which does not receive the intervention which is essential to determine whether the observed effects are due to exposure itself or other factors. Like they may receive placebo, they may receive the standard treatment, or maybe they are receiving no intervention and just the comparison group is there to observe the changes. Then is randomization. Participants are assigned to intervention or control groups by chance. So minimizing the selection bias and ensuring that groups are comparable at baseline means the though the randomization of samples are there, but they are not just taken as by chance. Okay, so they are carefully selected but randomly. There is masking. Masking reduces the observer and participant bias by concealing group assignment. Means if it is single blind, that means participants are unaware of their group. Double blind means both participants and investigators are unaware, while triple blind means data analysts are also blinded. Then we have defined outcome measures. Defined outcome measures means the study must specify clear and measurable health outcomes before any intervention begins. Then follow-up period should be there where participants are monitored over defined period of time and then the effect of intervention is being observed. Ethical oversight. All experimental studies must adhere to ethical principles. Then data collection and monitoring, that should also be done systematically. So what are the features of experimental epidemiology? So features are like manipulation of exposure, use of control and comparison groups, randomization, masking, perspective design, defined outcome measures, controlled conditions, high internal validity, ethical oversight, replicability, generalizability. These are all the features of this experimental method. Now we come to various types of experimental designs. So number one in this is RCT, that is randomized controlled trials, means in which the participants are randomly assigned to receive either the intervention or a control, that is a placebo. Placebo means we are telling them that this is a medicine, but it doesn't have any effect. Like होम्योपैथी में कहते हैं वन जो हम मीठी वाली लाइक टैबलेट्स लेते हैं, गोलियां लेते हैं स्मॉल, उसमें अगर मेडिसिन डाल दें तो देर मीन्स इट इज़ अ मेडिसिन, बट अगर उसमें कोई मेडिसिन नहीं पड़ती है तो देन इट इज़ वर्किंग एज अ प्लेसिबो. हम किसी बच्चों को या कुछ खिला भी सकते हैं कि वी आर गिविंग यू मेडिसिन, बट उसमें कुछ मेडिसिन है नहीं, देन इट इज़ एक्टिंग एज अ placebo, right? And then outcomes are compared between the groups. So key features are like gold standard for causal inference, minimizing the bias through randomization, and it is often used for drug or vaccine or behavioral interventions. Then is the field trial. Field trial means it is testing an intervention on a healthy population so that we can prevent the disease before it occurs. So it is focusing particularly on preventive measures. Then comes your community trials. Community trials means intervention is being tested, but it is tested on the entire community. Entire community or group means we are not bifurcating them, or we are not dividing them into healthy or unhealthy, but we are giving them as the entire community. So it is particularly used for environmental or behavioral interventions. Then comes your non-randomized, that is quasi experimental trial, where randomization is not there. We are using the statistical methods, or we are using some or the other method where we are carefully selecting our sample, right? Sample means whatever the intervention or whatever the um, testing has to be done, we are testing on particular people. So, for example, introducing new screening program in one hospital. So we are carefully selecting that hospital, and then we are implementing a new screening program in it. Next one is crossover design. Crossover design means participants receive both interventions in a sequence, but it is separated by a washout period to eliminate the carryover effects. Means one intervention is being tested, then there is a gap of some days or some months, then again the other intervention is being implemented. So each participant is working as their own control. So this is crossover design. Next is factorial design. Factorial design means we are testing two or more interventions simultaneously. Simultaneously means we are also able to detect the interaction effects between interventions, right? So these are certain designs which are considered under types of experimental methods. Next one is what are the steps of experimental research methods. So if we talk of steps or the procedure, so we have to first formulate the research question and hypothesis. We have to select the study population or the target population. We have to design an intervention. We have to see the control conditions, define the control group also. We have to choose the outcome measures, obtain ethical clearance. Ethical clearance means we have to ensure informed consent, confidentiality for the people, then randomization and blinding, implementing that intervention, then collecting the data, follow up, and then doing the analysis of that data which is collected. Then after the collection of data, we have to do the interpretation and reporting of that data, and we have to equally then share the findings with the stakeholders, policymakers, and scientific community. So these are 10 steps of the experimental design, or you can say the experimental research method. Now, what are the advantages? Advantages means we says that we have strong evidence for causality. Causality means randomization and control allow for clear attribution of outcomes. So no casual behavior is being considered for the sample or for the target population, or the individuals are not feeling any casual behavior like with them. Then minimization of bias and confounding, like random allocation and blinding reduces the selection bias. Perspective design is there, standardized conditions are there, direct evaluation and interventions can be done, quantitative measures of effect are done, and it also have ethical oversight. So these are the advantages of experimental research method. Then we have limitations. Limitations means ethical constraints are there, means some exposures cannot be assigned experimentally. Like we cannot have a group which should smoke or which should not smoke, right? Then high cost and resource intensity is there, limited generalizability, loss to follow up, complex logistics, time consuming method, and it is always not feasible. So this is all for your experimental research design.